Yeah, recording started.
Awesome. Well, greetings, everyone. Welcome to the National Institute on Aging Common Data Elements webinar. My name is Karen Anyanaho, and I'm the advisor for genomic data sharing with the National Institute on Aging, or NIA, in the Division of Neuroscience. I am joined today by my colleague from the NIA, Aaron Mazam, who I collaborated with in spearheading today's lecture. I'm also joined by another colleague, Raul Maston, who's providing IT support. Before we get started, I just wanted to go over a few housekeeping notes. One is that our audience is muted. We do have an in-meeting chat that has been disabled. However, there is a Q&A window that is enabled to solicit your questions throughout the presentation. So if you look at the bottom of your screen, you'll see that Q&A box, and we'll dive into a Q&A segment a little bit later to answer some of those questions. We also have captioning services provided by Adrena Atkins, and we can go ahead and get started. So I'm very pleased to welcome you all to our second monthly webinar lecture as part of our Common Data Elements speaker series, which began in April and will run through December 2024, featuring a dynamic lineup of speakers who will highlight best practices about standards and applications for optimizing data harmonization, as well as share insights about the usage of data resources, effective data management, and enhanced data analyses that their development and use are needed for common data elements and data harmonization to be fully realized. Our speaker series is designed to allow the NIA community to connect with Alzheimer's disease and Alzheimer's disease-related dementia and aging research and researchers who have expert knowledge in the field. At the same time, facilitate conversations and discussions about their resulting research findings, and also to provide a space to share other valuable information as it relates to common data elements, or CDEs. This afternoon, we are really delighted to have with us Dr. Mike Knauls as our featured speaker for today's webinar titled Systematic Data Harmonization Enabled by Generative Artificial Intelligence, Machine Learning, and Large Language Models. Dr. Knauls will discuss the application of generative artificial intelligence, machine learning, and large language models used for systematizing data harmonization. Dr. Knauls will also address inconsistencies and the lack of data standardization that hinder the ability for the utilization of data for building machine learning use cases. During this webinar, Dr. Knauls will also cover conventional approaches towards tackling some challenges, some of which I mentioned earlier, while also discussing how best to leverage the power of AI, ML, and LLM. Before we start, I'd like to share with you Dr. Knauls' professional background. He currently is a project lead for the Center for Alzheimer's and Related Dementia, known as CARD, which is a collaborative initiative of the NIA and the National Institute on Neurological Disorders and Stroke, supporting basic translational and clinical research on Alzheimer's disease and related dementia. Dr. Knauls leads the advanced analytics sector at CARD to advance open science to fight neurodegenerative diseases while building and deploying scalable advanced analytics systems. His research focuses on aspects of population genetics and inherent genetic aspects of population structure associated with neurodegenerative diseases. Dr. Knauls received his doctoral degree from Temple University with a focus on quantitative trait association methods and population genetic structure. Again, we are very pleased to have Dr. Knauls with us, and without further ado, I will now turn over to you, Dr. Knauls, for today's lecture.
Oh, thanks. It's been great working with you all to get this started, and thanks for having me. So I'm going to talk about a topic that's really near and dear to my heart, because when I was a GPP student at NIH, I remember having to harmonize data manually without all these awesome tools we have now. And that was right when I was also just getting done being a dishwasher and was now a real researcher. And I thought, wow, this is really hard work. Maybe I should go back to just being a dishwasher. So if we can make anything easier for the people that are, suffering harmonizing a bunch of disparate data sets, I hope we accomplish that with the work that we're doing at CARD. If we can move to the next slide, please, would be awesome. So I just want to give a quick disclosure. I am a consultant. I am not an FTE. There's a lot of the work that was done here and a lot of the technology developed was actually done to help the Center for Alzheimer's related dementias under a competitive contract to support data science and advanced analytics. Next slide, please. So, I think this is really the big picture, and it's Friday afternoon, so if people start zoning out at 4:15 p.m., it's totally cool, because these are the kind of two big take-home points in advance. This is the TLDR. So AI readiness is huge. This is hugely important at NIH, but it's not really a new concept. It's the basic premise is like, you should have the same column names, the same content, roughly the same dynamic range for every variable. It's not big, but it's growing in importance, particularly in the space of things like GDPR and other data silos that we now operate in more of a meta-analytic and federated framework than we do as a centralizing framework. Of course, centralizing data is always wonderful, but it's really important. So we're going to talk also about human in the loop, and this is multiple levels. There's a technical level, and then there's a conceptual level of human in the loop. Specifically for large language models, you save a lot of time and effort. You can automate tedious stuff. And that's what a lot of this talk will be about, is really automating tedious things, right? Anybody who's ever harmonized a data set gets weird abbreviations. They have no idea what they are, and there's no documentation, and they have to look those up. LLMs are great for that, because LLMs are really good at looking things up on the internet for you. And using machine learning, really basic ML like elastic nets, you can really start matching data sets that aren't one-to-one matchable, right? Using fuzzy matching and things like that to help save time and flag what needs this human to curate data, right? So if you can automatically cut your job size down by 80% because the LLM can do things pretty good, and then have a human spend the rest of the 20% of the time for the job auditing these results from the LLM, then you've saved yourself a huge amount of work, and sped up the project and systematized the project for data harmonization much more. But we'll get into the details of this. This is just in case people want to clock out early and start and go about their weekend. This is the core of what we're going to talk about, just without the details. So person important, AI is a great tool. We're getting AI ready for larger, federated analyses, but let's get into the details now. Next slide, please. So this is really the evolution of biomedical data, and this is why data harmonization is so important. We have these, easy, tiny studies, these family-based studies, for example, of one SNP that you could do on an Excel file or something like that. If you use Excel, I have no idea how to use it, but it's kilobyte sized. Then you have, in the 2000s, you have this earliest genome-wide association studies appear, and this is like, you could do this on a laptop, right? It's a few megabytes of data, some clinical data, one site, one data set, right? In the 2010s, we really started having to think about sample size and how we could work across studies, right? And what this does is this kind of has you working across data silos, doing meta analytics. And this is when the importance of common data elements and what people now call AI readiness or data harmonization really comes together. This is a practical aspect of it, where you have meta analytic tools that need to be deployed across silos, so that you can make sure that you're analyzing the same data point and meta-analyzing or combining results from blinded studies in the same way, using the same data. This is a laptop size, or maybe a small cluster, gigabytes of data at the most, right? 2015, we hit this transitional phase where data got really, really large, particularly in genomics and EHR data, which is what a lot of our team focuses on. This is, imputation sequencing level. This is whole genome sequencing, GitHub to share tools across data silos, cluster computing, some data centralization, but really it was terabytes data dispersed across silos. And we had to find ways to meta-analyze this. Once again, the need for common data elements and data standardization, just even having the same column names that you're going to analyze across these different studies, is of utmost importance, right? Simple things like that. And there are a number of statistical checks that are time consuming, that you can make sure that things are in the same dynamic range, that, variable A in one data set is still measured in the same units as variable B in the other. But it's better to really use common data elements and that structure to get that harmonization done upfront across all the studies. Finally, we are now in this kind of era of like centralized research and AI readiness at the forefront of everybody's minds. And this is the idea of either bringing everyone's data into the same cloud platform, but sometimes that's not possible. So prospectively federating studies, and this is something that like the GP2 initiative and You know, a lot of work that's being done in the CRISPy pilot and stuff like that is really focused on, and it's been wonderful to collaborate with them on how to bring global, you know, hundreds of study centers together to have uniform data for genomics and for clinical data. It's been at longitudinal clinical data that it's been wonderful to work in this kind of data discovery and data harmonization aspect that really helps people come together as a research community. And I think, you know, CDEs sound boring to many people, but that's how you build a research community that works together, right? If you make it easy for people to interoperate in one way or another. So things are easier now because people are actually aware of this and it's been socialized. And, you know, we have great cloud platforms now, right? We have our card, we use the Alzheimer's disease data workbench, and we use the Verily workbench, and they've been exceptional for a variety of different projects and different tools. And there's many more options out there, but bringing people together, unified compute, unified data structures, it's awesome. So let's talk about how AI makes that a little easier. Next slide, please. So you get these federated data sets, right? And we've all had these problems. These are some of the problems that we're trying to address, right? Redundant data storage, you know, having one copy of note of all the data and making them interoperate, you know, not having to reinvent the wheel. These are all things that we can leverage CDEs and AI to help make easier and save money and time, so you can be better spent innovating. Next slide, please. So we've really tried to boil this down, you know, because I'm not an exciting person. So I worked a lot in kitchens and I think of everything as a recipe. So this is kind of just our overall recipe for sharing brain relevant data sets at CARD. And you'll notice that 2 out of 4 of these boxes that are highlighted on the right have CDEs in the header. That's how important these are to really bringing data sets together and sharing data sets effectively, because we're trying to establish global community standards and trying to interoperate with other groups like the NLM and other and potentially other teams that have really worked on setting these standards, so that we can all work together across domains, across expertises in a fluid and safe way without having to reinvent the wheel constantly. Nobody wants to clean the same data set as their collaborator did, you know. Next slide, please. So we have too many silos, we have too many repositories, and this really increases activation energy. So one thing we did to catalog both CDEs and data sets themselves based on these CDEs is to build a lightweight tool that lets you browse data sets and lets you browse thousands of what we call generative common data elements. So data elements that we've taken from different data dictionaries and different study headers and even column headers from data sets that had no explanation. Use generative AI to basically build lingua franca for data dictionaries, and manually audited many of those to see if they needed to be changed or need to be tweaked, because right, sometimes AI will lie to you, but one thing it's very good at is looking things up on the internet given a defined context, such as aging research or, you know, brain research or neurodegenerative disease. Next slide, please. So we built a tool called Diver, and Diver is this data inventory and verification research environment. It's a really straightforward tool for data discovery and harmonization with awesome work from the teams such as Hirotaka, Shannon, Liesel, Alan, Syed, and Nicole. And it's been awesome to work on, really kind of working within the context of fair and discoverable data to be more efficient, to be more interoperable. We want to build this one data dictionary to rule them all. Eventually, that was, you know, a quote of Lord of the Rings, which I actually just saw for the first time recently because everybody made fun of me in the lab for never having seen it, but using that quote. And then fuzzy matching and elastic nets to kind of browse across data sets to make sure that they're in line with these common data elements that we've been assembling. And I'll talk more about how data discovery and common data elements should go hand in hand in your FAIR proposals. Next slide, please. So what we're doing is just essentially cataloging, harmonizing, and tracking common data types and issues, building data dictionaries, and defining variable harmonization. For certain things that you'll see later, we use this tooling to build also common data elements for things that don't exist yet. Right now, one of my favorite projects we're working on is a digital pathology project that these variables actually don't exist. So we're working with subject matter experts to set the standard and letting the AI kind of infer what these standards should be for us, and then we tweak it, right? This is part of that human loop idea. The AI is not going to solve all your problems, but it's a great tool in your toolbox. Next slide, please. So we've been really busy kind of working across all of these data sets at CARD, and this is like drinking from the fire hose. So we need to build tools that will allow us to both locate these data sets, parse these data sets, and work across these data sets without having someone do the manual labor because it was just too much. So this is essentially the impetus for why we did this. And these are all great resources. Shout outs to everyone who put these out in the public domain. Next slide, please. So this is a little video of kind of how the Diver Google Form query tool works. So we have multiple flavors of this data discovery tool going out that catalogs data types and data modalities, but also it's a video. I'm sorry, if somebody could just hit play on that one, that would be awesome. Okay, so the video is not working apparently, but there is the next slide. There's just kind of like an idea of the drop down. So this is a browser for standardized metadata, right? Because metadata for studies is also basically a common data element. You need to know who the contact is, where it is, what the study ID is, all of these things, because we're talking about study interoperability and AI readiness. And what that means is that we need to be able to rapidly find, say, studies that have genotyping data and Alzheimer's disease across all the repositories that we have access to. So it's really exciting that we can just kind of log in a secure site that's managed with two-factor, and you can just browse all of the data within CARD that is available based on both CDEs and data modalities. It's been really cool for us, particularly for as a future proofing for the upcoming summer when we have like 20 summer students visiting and they're all going to ask what kind of data is, right? That's a lot of emails. So if this tool can cut down on those emails, we've done something right. And if they can find the data elements and data modalities that they're interested in, then that's great. Next slide, please. We also made a chat interface to this because, it's fun to just ask questions like list all the studies with Alzheimer's and clinical metadata. And then it will give you an idea of, what's been cataloged, and all of the cataloging is just a very brief Google form per data set. And, it's nice for people who don't want to write code and also don't like drop-down menus. Next slide, please. So now getting into the other use of AI past just cataloging data is automating the tedious parts of data science, right? Everybody on the call that writes code probably agrees that 70% of our jobs are just making sure that data is okay most days, right? We've all spent like weeks cleaning data sets just to do 3 logistic regressions and call it a day. So if we can do things that make that easier, then why not use all the tools available? So context-aware AI has been key to this for us, right? We want this kind of research assistant in your browser. Next slide, please. So the team that did Diver evolved into building these generative CDEs. And this is really important because this complements a lot of the work that we've been doing. And it really lets us save so much time. We'll get to the time savings in the next slide, I think. So we're leveraging heavily engineered prompts to get to the idea that if you have something sparse or something messy, we can eventually make a data dictionary out of it. It just might take an AI and sometimes a human. And this can really complement or accelerate our efforts at the NIH and beyond, because then we can build a standard for new variables and new data types like spatial transcriptomics and digipathology that are coming out now and really coming to the forefront that haven't been thought about before. Next slide, please. So this is how we think about sparse data harmonization made easy, right? You get these tabular data in various formats from disparate data silos. The AI ingests the data and builds these CDE parameters for each input. And then we deploy Python scripts that check, do these match with previously harmonized data sets or data types or common data elements, so we don't have to keep doing work over and over. We generate quality reports based on the internal characteristics of data after the CDEs have been matched. And then federated analyses can now begin once these have been matched across data sets. That's usually where there is some human in the loop, because we are flagging what variables might need to be altered if they're outside of the scale, right? If you have a variable that's an order of magnitude larger than it is in the CDE boundaries, then we know that variable needs to in an automated fashion, we can flag that that variable needs to be adjusted at the study level and should not be analyzed in that context, which is something that saves a huge amount of time if you ever had to do this manually. The coolest thing is that this is a learning system, right? And because of the matching, once a CDE is built, we don't need to rebuild it. We can just track the aliases for that CD and then match those on new data sets as they come out, or extant data sets as they're being harmonized. And this all gets easier and easier every time we fire it up. Next slide, please. So this is kind of the meme of where it started and where it went. We were analyzing data set and I had no idea what 2min WT was as the column header. And I was like, oh, what is that? And I was Googling and it came with a bunch of other things. And I was like, well, GPTs are very good at context. So let's figure out what that is. And it did a pretty good job. And then 4,000 some lines of code later or whatever, it went to a Python script that has with heavy prompt engineering that then makes this into a tabular data set based on a variety of inputs that are textual, from a data dictionary that has missing columns to just the headers of a file. We try to do the best we can to match those to either existing CDs that we've cataloged internally or build brand new de novo CDs using this context-based LLM. Next slide, please. So this is what you can see here is from a couple weeks ago. Generally, we get about an 85% hit rate across a lot of these different data sets, where we can build a CD from a header or from an existing data set where no edits need to be made to any of the fields that are going into this massive data dictionary of tens of thousands of potential variables in a highly parallelized and cost-efficient code that is PHI secure. We're only dealing with metadata that's abstracted, but there's no participant level data going into this. So it's really saving us time just by flagging what might not work well for data harmonization, right? We can go through here and we can have if you're 85% on the mark, it's way easier to edit these CDEs and these data dictionaries than building de novo entries, right? You've just cut your number of PubMed searches and Google searches by an order of magnitude for the most part. It does take the impact of a CDE like Alan Long and Hiro Takawaki, Shannon Ballard. They do amazing work kind of revising these and doing the due diligence on these, because these, you can't trust the AI, but it does pretty good work when it's right. Next slide, please. So this is kind of what's going on. This is just a peek at like some of the variables and how we format this. We really focus on categorical variables, continuous variables, and binary variables. We tend to use the AI to reauthor ordinal variables into categorical variables because that is somewhat tricky to do in a standardized way across different sites. But you can see that we're just basically making big tables with lots of aliases for the variables. So we have an easier time matching these across studies because study abbreviations, aliases, things like that all change pretty rapidly across publications and data sets. Next slide, please. So we also are in the process of refining what we're calling the interoperability score. This is a per study variable, and we're doing really simple machine learning applications of ElasticNet to match variables across data sets to the data dictionaries, and then calculating how interoperable these data sets are on the variable level, on the CDE level. This includes calculating a missingness rate threshold, tolerance threshold, so a bracketed out min max for continuous variables, a binary coding, so looking to see if there's a huge frequency difference across that past what's expected for any binary variables, right? You don't want something that's really, really common in one data set and really rare in another. That could be a coding issue, right? They could be flipping what's 0 and what's 1, for example, or it could be a disease-specific data set, but flagging that provides valuable information to the data scientist that is working across these data sets, and that knowing creates better science and saves them a lot of headaches in the future. And finally, fuzzy matching for these categorical variables. We want to see how much content we can match across different categorical variables at a threshold of 0.8 likelihood of a match or above. That gives us an idea of if the coding of these categorical variables is the same. And Alan Long has been grinding this out really intensely across 8 massive data sets, I think, this week. So we should have a really nice workflow for this to release soon. Next slide, please. So why does this CDE platform accelerate research? So, right, 85% of the CDEs don't need editing. This is, this works extremely well for well-known abbreviations, common variables, ICD codes, 9 or 10. We're really lucky to have CDEs that SMEs that spend a lot of time evaluating this. We think we can work with other NIH data harmonization methods to really accelerate their work, as opposed to, having a very overworked graduate student suffering somewhere doing this, because I was that person at one point in my life. This also, because there is no PHI in this or anything like that, except for when we distribute the interoperability score code, it allows us to maintain the data silos safety, and it's really just ends up at the end of the day flagging outliers is what it is, right? So that the site analysts have an easier time harmonizing data, and it's context aware, so we can deploy this workflow. It's the biggest test has been in healthcare, but I've been playing around with it in finance and climate change, and there's potential there too, right? What if one day NLM worked with us and we made a data dictionary about everything? Like, that would be cool. I don't think that's super realistic, but it would be theoretically possible. Next slide, please. So I'll talk about putting this into practice now, because nobody likes just to be talked about pipelines that are being developed and concepts. It's really putting this in practice. So it's just so I'll give you 3 examples of how we've leveraged this to really make our lives a lot easier at CARD. So next slide, please. So this is one of my favorite projects we worked on. This is the Digital Pathology Research Sandbox. This is going across 15 partner academic and nonprofit institutions to build a community for digital pathology research, and lay the foundations for a spatial genomics community and genetics community, as well as traditional pathology. It's been super exciting. We're focused on tabular data, all harmonized to the same standard, so that we can then, proof concept for the end of the year is to run a GWAS of these these digital pathology markers in the brain. And it's been super exciting, but we had to, we're running a very tight schedule, as you can see. So we had to build our first wave of CDEs very quickly, in less than 2 weeks is what we gave ourselves. Next slide, please. So we employed a number of sparse data dictionaries that we got across different collaborative centers, but also just essentially writing variable names and brief descriptions that we thought, running it through this AI pipeline, tagging them to existing NLM CDs if they existed. They existed for, I think, roughly 70% of the data, and then building in things like the content and all that, so that we could then review these as a team. And all the sites were quite happy with the 267 built so far. And now we're just adding new novel data elements that are pathology specific, that don't really exist yet in the public domain, but hopefully they will. And we're looking forward to sharing these with the greater community very soon. But we did this from point zero across 15 centers, plus multiple data dictionaries in hand, synthesize this in less than 2 weeks. So just leveraging the formatting and munging that the AI can do pretty well, given the context. We're very happy with the results, and it's saved us a huge amount of time. Next slide, please. So federated learning is another thing that we really enjoyed exploring, and the people highlighted in the neon blue box on the left, such as Ben, Mary, Anant, Dan, Paul, Andy Singleton, Jimeng, and Faraz. This is really light years ahead of where I thought we would be, being able to really work around different data silos to build models that function better than centralized models on a single data set. It's been wonderful to see, right? This is abstraction of the statistics and machine learning approaches that help with your generating predictive text on your phone, right? So learning across silos, abstracting the data in a PHI secure way, and building a centralized model that can then be tested on withheld data sets. And it shows that this is possible as long as you have harmonized data to the same CDEs. So it was extremely important for us to make sure that all the silos that we were testing on, even the simulated data, everything was kind of lining up in this CD paradigm that we've been talking about and leveraging the tools that we've been doing for this. And the paper's there, Danik et al. It's wonderful. I'm really proud to have worked with all these amazing people on this. Next slide, please. So also, Card AI is something that we've been building. This is a massive collaborative effort. You can see all the names on the bottom. It's really fun to have a online research assistant, and the research assistant itself will use some CDEs to describe associations across different studies, which is nice. I mean, we have gene level associations that can pull up really easily. If you go to the next slide, please. It synthesizes all of these data points and brings all of these data sets together. But the only way it can really synthesize these data sets is if there are common terms that are reflected in harmonized data. So the data engineering team that's been working on this has been amazing at using standard nomenclature for everything, and that allows these SQL queries to work well, right? So this is a proof of AI readiness, and I'm really excited for Faraz and the team that's running with this to be showcasing this to the NIH community very soon. So it shows that not only are we dealing with CDEs on the clinical standpoint, but we should start considering genomic and genetic data to have its own CDEs, right? How do we get these data sets interoperable? Do we want to use RSIDs? Do we use ensemble IDs? These are all things that need to be mapped out before you start a study of this scale. And it shows that there are multiple levels to CDEs and AI readiness that are not generally considered by everybody. But data's growing, data's getting deeper. We should start working in that direction, right? We can all agree that a fork is a fork in the English language. We should do that with science. Next slide, please. So it's really been wonderful to work at CARD. There's a massive number of people that were involved in all these projects, Corey Wellers, Rose Abbas, Maya Wiley, Nicholas Johnson, Kendall Jensen, Elise Marsan, Sarah Bandres, Andy Singleton, Cornelius Brown, Kim Billingsley. These are a bunch of people that have been working behind the scenes, and Andy Henry, Hampton Leonard, Chelsea Alvarado. There's a huge list that keeps going on and on. Pilar Alvarez, Mary Macarius. Yeah, it's I'm going to just mentally try and picture where everyone's sitting at CARD right now to say thanks, because this is all a huge community effort, and we're making common data elements based on community data to bring communities together, and maybe it should just be called community data elements. I don't know, it's pretty bad dad joke, but it is what it is.
Wow, thank you so much, Dr. Knowles. That was a really insightful and very thought-provoking lecture. Greatly appreciated you sharing this presentation, especially your expert insights about systematizing data harmonization, as well as effective approaches needed towards enabling artificial intelligence, machine learning, and large language models. So we do appreciate that. We actually have quite a stimulated audience who has been engaged throughout your talk. And so at this time, we want to turn now to our Q&A segment so that we can engage our audience. First, thank you so much for the questions that you sent in throughout the presentation. We do have an additional 5 minutes to add to our Q&A segment. So at this time, I will go ahead and dive in. One question I have for you, Dr. Knowles, is would you suggest that artificial intelligence is simply a tool in the toolbox per se, and that it is not going to necessarily fix everything? And if so, depending on your question, how extensible would it be to, for example, incorporate other data modalities across different industries?
Yeah, I think it's pretty accessible. I mean, I always think of like having a toolbox, right? And then thinking kind of AI can do a hell of a lot of work really fast, but it can also break stuff and hallucinate pretty easy. Like, think of AI as a Sawzall. I don't know if you know what that is, it's like a demolition saw. You know, it does a lot of work really fast, but you can mess things up with it. Keep that right next to like your logistic regression hammer.
Awesome, awesome. No, that's quite interesting. We actually have quite a few questions. So I'm just gonna go through each of them so that I can engage you with the audience. Who again, we really appreciate you all sending your questions in. So one of our audience members mentioned your talk about CDEs with genomic data. They've asked how common are those metrics? Things such as RNA sequence, have various processing biases that could confound, for example, cross data analyses. Is this something that you've sort of adapted into these various models? And if so, could you provide us with some insights there?
Yeah, so we try to use the CDE components for the CDE state of mind for everything, right? It should, for, I made most of my career doing meta-analyses of like, hundreds of thousands of samples across, 20 different data sets that, half of which I never saw. So the idea of making sure that every gene is named the same, that every counts table is on the same scale, because you get things like when you do a regression, the betas will be the different size, and that will throw everything off, right? That will literally throw the stats off. You can quantify that with statistics like I-squared, but you can also address issues like that with, unsupervised learning, such as principal components, right? Adjusting for those as covariates in your models when you combine data sets, if you're not going to meta-analyze them. The other aspect would be kind of thinking about latent spaces, essentially looking at the white space between clusters of things and seeing what is pushing those or driving those, down to simple solutions like you might just have a batch issue. You might just have a few bad batches that might need to be adjusted for as covariates. I think all of these are things that data scientists need to explore. And at CARD, we're really doing hard work, particularly on the genomic side, to automate that and automate these standard pipelines. We've rolled out some really cool pipelines for single cell sequencing, for transcriptomics, as well as some ATAC-seq work. And then we've just put up a web server for mass spec proteomics so that people can harmonize their data by generating it uniformly so that it is to at least some degree interoperable. Of course, you'll always be doing things like, PCA to see if, and color coding by study to see if one study is completely on the other side of the map from another, but that's just called data due diligence, right? That's what everybody has to do.
No, yeah,
We try
absolutely.
to make that not as impactful though.
Totally. Thank you for that insight. I also appreciate earlier in your talk where you were describing, how students, for example, fellows are utilizing the Diver tool for data discovery.
When they show up, we're just rolling it out now.
Yeah, actually, that was a question that I had. I think another audience member had as well about, some of those efforts that are being made to actually bring the students to the tools. Are there any perhaps best practices or lessons learned that you could share with us that you take into account to mitigate any challenges with using tools that could arise, for example, from attempts to harmonize poor quality data, an additional time that is used to clean the data?
I think you just have to be cautious, and this varies from center to center and lab to lab and institute to institute, how much of the knowledge base is there. I mean, I would love it, my life dream, probably because I've spent way too many times sitting in a dark office harmonizing data sets, would be if we just came up with universal global standards for everything. And there's some places like the NIH or beyond that could do that. I mean, we're trying to do that on a small scale at CARD. It's just there's a CARD data standard that we hold things to.
That's great. There is actually another sort of follow-up question from another audience member who's asking, for tools like Diver, as they get more complex, do you foresee more human, work going on to enable the harmonization of different data types, or even accommodating the field being already supported as this evolves?
I'm sorry, that's not super clear. So like, I think we're getting better tools all the time, but then also some old tools are great. Like we benchmarked some pretty modern fuzzy matching workflows against like ElasticNet, BM25 ElasticNet, and shout out to Owen Bianchi. He's done a great job, being like, well, BM25 just works amazing. It's things like that are important. Just testing everything and having really good people on your team. It's awesome for me to walk into the room and probably think I'm the dumbest person at CARD. It's like these people are surrounding yourself with these great people that can make those decisions. You'll always need engineers. I think the AI is not going to swap out the engineers, and you'll always need subject matter experts. But what our goal is with these more complicated tools is to make the tedious parts of their jobs easier so that they can innovate and build better tools or do these due diligence things where they're optimizing things like what Owen did.
Yeah, that's really helpful. Thank you. You've actually answered the question that came into the chat. On that note, and thank you for sharing how it is truly important when you do have a collaborative team to help really streamline many of these efforts that you nicely described. Another question that I have, I know in some cases, if training data that's used for large language models could include biased or unrepresentative samples, the model could inevitably learn or perpetuate biases. These could include gender, race, and cultural biases. One question I have, are there any lessons learned that you could share about how to address mitigating biases in large language models?
Yeah, I mean, that's really tough. So, I mean, I think this is where ideas like RAG and tuning models to important data sets to prioritize those is really exciting. I mean, it's also we just finished a systematic review with NLM, right? And it's the paucity of data, of diverse data out there that really, those are the gaps that we're trying to fill at CARD, right? We saw that less than 10% of all genomically quantified data was diverse, and that's something that we're working really hard to address, so that models are more generalizable, right? You have to have the data to train. So sometimes data generation is actually like the important part, getting enough data.
Absolutely, I couldn't agree with you more. One question we have while we're still speaking about the Diver tool that's from our audience, is the tool accessible to data end users outside of the CARD ecosystem in terms of accessibility? So
Christine, that's a great question. Thanks for taking your Friday afternoon to listen to me talk. Sorry if it was boring. It's not right now. We're still beta testing it. That's why I said we're going to find out how exactly good it works with when the students start showing up. We have an excellent training and outreach team at CARD that's going to help us help them, and then we'll get the data on that. And then it is not difficult to roll out something like Diver or Diver itself at different sites once you have the workflow down. It would be super exciting if we could work with other teams that have different institutional access controls and different data sets and different interests to build to build something for them and have them crosstalk about what, sharing on a very superficial metadata level, who is working on what. You could actually find out based on the metadata available about the data sets.
Yeah, thank you very much, Dr. Knowles, and thank you for that question from our audience. We do have another question, sort of switching into the real world data. One question is being asked on how can these tools be effectively deployed to longitudinal studies, for example, heterogeneous visit schedules collected out of an EHR or registry data. Could you speak on that?
Yeah, so that's something we've been addressing with the DigiPath Research Sandbox, and we've been just delimiting. There's always paired variables we've been building in these paired variables for longitudinal data, in which it's delimited in the first variable. So you have the value, and then below that is the date of the test.
Awesome, Dr. Knowles. We also have another.
Dates are really hard though. We try to make sure that in all of our standards, the default standard that the LLM and that us prefers is the Unix standard, but some studies only have time to event, so we have to approximate those when cleaning the data.
Actually, there's another question in that same vein that it may not appear in the Q&A, but we're getting it. So thank you. I actually wanted to sort of punt this next question to our Director of the Office of Data Resources and Analytics within the Division of Behavioral and Social Research, Dr. Partha Bhattacharya. Could you please ask your question?
Thanks. Mike, I'm an economist. So here is a related question, right? We have seen manual curation of longitudinal data. We have seen manual curation not being done properly for longitudinal data, where the variables are labeled differently depending on the year you collect. So when you do the fixes, right, using the machine, can you give us an idea of time savings, cost savings, and the marginal return, right? It's that's what I'm
Yeah.
seeing here. And can you quantify that for us? Again, it's hard. I'm not saying give me all the numbers, but I think with 87 people on, I'd like them to hear like
Yeah.
what is the ROI on something like
So we see an order of magnitude speed up of this because you're able to you can review it really fast, right? I mean, I don't know if anybody here has tried to build a CD, but you have to look up a lot of different things and read a lot of articles. Whereas if you get a subject matter expert that knows what they're doing, they can chew through some CDEs pretty quick. I think the don't hold me to this number, but I think the fastest we've ever seen is like 4,000 in a week. And that was like lights out, sitting, only one light, sitting at your desk suffering over the holidays with no emails. But like we did 256 or 267 with a lot of back and forth via email between people that didn't like the names of certain variables and stuff like that in less than 2 weeks. And that's still pretty good considering if you look at a lot of other initiatives that have been around for years and, have delivered 200 adjudicated CDs. So, collaboratively, we can do a lot as a community. And I think tools like this are here to help everybody. So let's all work together to make everybody's life a little bit easier. Nobody wants to be sitting in the dark room Googling what is the unit of measure for hemoglobin A1C, right? When the AI can spit that out
we
pretty
shouldn't be
quick.
doing that today, and I'm afraid
It happens a lot.
Still building that, right? So
Yeah.
Thanks.
Why is this measured in deciliters versus picoliters or whatever? Like I don't know. That stuff sucks. It's hard to do. It's time consuming. And those people who generally do that also have strong analytics skill sets. So their time could probably be better used building a model than Google searching or PubMed searching. Just that's my personal point of view as having been that person for quite some time.
And thank you so much, Dr. Bhattacharya, for that question. And thank you, Dr. Knowles, for these really great insights. You talked earlier about efforts taken at CARD to deploy data dictionaries gathered from collaborative centers and other sources to add new novel data elements, for example, within a relatively short period of time. An audience member asked, how did you build the CDEs in just 2 weeks, and what processes you use? I know that you mentioned earlier.
And we got a bunch of data dictionaries, and then we did cut and pasting the lists, and then we had the AI go through it, and then we just went through and manually audited that afterwards and made a Google sheet, and then all the collaborators said what they didn't like that it did and what they liked that it did. It's the coding and the other parts of it, the hardware, the software part of it is difficult, but like there's also that collaborative community part that is really exciting, but also time consuming, but really rewarding at the end.
Yeah, and that's always awesome when you have that rewarding experience at the end. And again, takes a really collaborative team. So thank you, Dr. Knowles, for that.
When people that you really respect in science go, wow, that was pretty good and really fast, it makes your day.
Indeed, I'm
pretty happy about that kind of thing.
Absolutely. Thank you very much for sharing. We have a few more questions before we end our Q&A segment. One question I wanted to ask, actually, let me go to this question from one of our audience members. They're interested in the DigiPath CDE, if I'm saying that correctly, with the NeuroBank, with the NeuroBiobank. They want to know, could you share that outside of this forum? They're asking, or they're mentioning it would be great to share those CDEs with the NIH NeuroBiobank sites. Is that possible?
Yeah, Christine, just drop me an email and we can, it's actually probably good to get a coffee and chat about this.
Easy enough.
Yes, no worries. It's a Google Sheet now, so anybody can check it out.
Thank you, Dr. Knowles. During the talk, you had also mentioned, many silos exist, and I know many of us in the data science space are aware that this is that there's a lot of truth in that. With repositories of biomedical and clinical research, for example. You also nicely described how CARD, built a generative component or a catalog of CDEs, which takes into account the various data dictionaries and studies that contain data or even metadata to enable data discovery and harmonization with the Diver tool, at least. I've actually really enjoyed you talking. Sorry. Yeah, no, I think I can say myself and many of our audience members enjoy you talking about the Diver tool and its ability to enable, how do you define data harmonization and then build CDEs that may not exist yet, right?
Yeah.
Wanted to know, how do you account for, or if there are any other varying factors or solutions? Are there any data harmonization solutions that you've not yet spoken about in the Diver pipeline that could account for, differing factors as you continue to build those CDEs, such as variety or volume of sources that you use?
Yeah, I mean, like certain things, longitudinal data is tricky. Medications are tricky, particularly medications across different countries because they have different names and different abbreviations that aren't indexed as well. It's been really, really good. I mean, with ICDs and stuff like that, just kind of getting the rough frequencies of those things. We have to think about it in two separate things, right? You have this aspect of data discovery, which is like the drop down menu thing you saw, and then the CDE thing, which does not have that same user friendliness, but it hopefully will very soon, that the rest of Diver has. But we're really working hard on that at CARD, and right now it's internal tooling that we're using to make these kind of just spreadsheets basically that communities audit. But it will be there hopefully. Over the summer it will be there. It's just we're working really, really hard to get this all done, and it's not the easiest engineering, to say the least. But being able to find your data is great, and being able to cross-reference CDs is awesome. We're just doing this because as we're building new data types, they need these CDEs as well, and I didn't want us to have to suffer doing this. But also there's a bunch of standards that are out there that people name things different things, and you have to start matching across those. And I think that's one thing that we did a very good job, with this human in the loop situation that we've had of matching these CDEs and then kind of smoothing them out using the AI.
Yeah, excellent. In fact, that was something that was majorly discussed over the past couple of weeks at the Global Alliance for Genomics and Health, or GA4GH Connect session. So appreciate you actually highlighting the importance of that. It's been a very popular topic. We still have time for maybe a couple more questions, and then we'll dive into a poll for the audience. We want to make sure we learn from you what you enjoyed most about today's lecture and other topics in the future that we might want to consider. And so, to you, Dr. Knowles, bringing global data and studies together to enable genomic and clinical data analyses and discoveries, what you discussed earlier, to allow research communities to come together, to build from data harmonization. During your talk, especially you mentioning how important it is not only bring people together, but in a unified structure through AI applications. Could you share with us how else AI and CDEs could be leveraged to spur innovation in scientific discovery that has not yet been seen, with the ability to share data and interoperate safely?
Yeah, that's a big question. I mean, I think with some of the work that Ben Danik and everybody who I shouted out on that slide previously, we can start learning across silos. We can start building predictive models for disease or drug efficacy across these massive healthcare systems, right? I mean, that's essentially right. If you think about all these biobanks out there, they're national scale healthcare systems, but that data usually can't leave anywhere. So if everybody adheres to a similar standard globally, we've just saved the entire research community time, because they don't have to guess, right? Like simple things like 2min WT for 2-minute walk time versus 2m WT, right? If you're just writing code, you just have a missing variable right there because you've lost 2 letters in an abbreviation for a column header. It's those things, just avoiding those dumb things is really key. I mean, I'm sure there's a bunch of people like myself who spent most of their life writing code, and they've lost days of their lives to a couple spaces that somebody put in a file by accident, like maybe you did. It's standardization and error proofing things is part of this whole process that makes people's lives easier.
Yeah, I couldn't agree with you more. That was a really, I think, a great explanation. So thank you for that. And I apologize to the audience for any questions that we may not cover today. However, we will make sure that we do get back to you, and as well as enable access to the recording for today's lecture. But I'll end with, before we actually open up our poll to the audience, knowing that data harmonization is really a really important aspect in building not just CDEs, but enabling the deployment of the tools that you talked about, artificial intelligence, machine learning, large language models that facilitate data captures. from various sources, that help clear away errors, for example, presenting results accurately, right, to build this body of knowledge that allows data end users to effectively and efficiently access, crucial knowledge to facilitate informed decision making for scientific discovery. I thought was truly valuable in your talk. So thank you so much. I do want to thank you, Dr. Knowles, greatly for your expertise, for engaging us throughout the last 45 minutes or so, and for sharing your insights during today's webinar, which is highly valuable in advancing the field of common data elements, as well as data harmonization, ensuring scientific progress, as well as supporting NIA's mission to conduct genetic, biological, clinical, behavioral, social, and economic research on AD, ADRD, and aging. So thank you very, very much. And now before we close, our audience members, you do see we've launched 2 questions in our poll that we hope that you'd have some time to answer. One is, what aspect of Dr. Knowles' lecture was most interesting? Many of you expressed that in the chat or in our Q&A window, but feel free to share those thoughts in that question. And then the second is, what other topics would you like to see in a future webinar? I mentioned earlier at the start of the lecture that through December of this year, we will have other monthly webinars for the CDE speaker series featuring other dynamic speakers with a volume of expertise. And we'd love to hear your ideas on what other topics you might want to see throughout the rest of the year, and also maybe in future webinars that we may hold in subsequent calendar years. With that, I wanted to thank all of my colleagues, Ms. Aram Azam, Raul Mastan, our CART captioner, Adrena Watkins, the director, as I mentioned, of ODRA earlier, who hopped on, Dr. Partha Bhattacharya, and to all of you, and especially to our speaker, Dr. Knowles, for taking the time out of your busy Friday afternoon to, participate in today's lecture. We will share the recording at the end, and any questions that were not answered, we will be sure to get back to you via email. With that, again, thank you all for your time today attending today's lecture by Dr. Knowles. Have a great rest of your day.
Really appreciate all the help, and even just the help of getting the slides up, since Zoom hates me, apparently. Thanks a lot, everybody. And thanks for the
You made it
questions.
easy, Dr. Knowles. You did. Thank you all.
Good one.
Thanks.
Bye-bye.